Hello all. This is a re-recording of the session 1. There was an error in the recording session. So this will be re-recorded again for posting on the YouTube channel. So in the session 1, we first introduced the course, that NPTEL host pathogen interactions. The course instructor is Professor Himanshu Kumar from ICER, and I'm Sumati Raviraj, PhD student from University of Hyderabad. So the course is of 12 weeks and the start date is 20th of January. Enrollment ends on 3rd of February. Exam date is 4th May and exam registration ends by 14th Feb. So about the course, this course introduces about the fundamental aspects of host defense system, that is human and mouse host defense system, and also discusses how our immune system develops immunity against foreign living and non-living entities. This course will be excellent for bachelor's, master's, or research level students from different disciplines of biological sciences, uh, biotechnology, chemical sciences, and medical sciences to understand the basics of the host defense system. So the prerequisites for this course is a bachelor's degree in biological sciences with some knowledge in biochemistry, molecular biology, and animal physiology. The basics of these branches will help you understand the course much better. So coming to the course layout, this is a 12-week course. So in the week 1, it covers history of development of the immunology field, branches of immunology, and an introduction to the immune system, overall introduction of the immune organs. Week 2 starts with immune organs, that is thymus and bone marrow, immune organs like spleen, lymph node, malt, uh, and galt, cells of immune system, including hematopoiesis and usage of early stem cells in transgenesis. Week 3, cells of immune system. This includes development of transgenic animals for immunological studies, applications of transgenic animals for immunological studies, and then various immune cells and its role in the host defense and immunopathogenesis. Week 4, basics of cytokines and other immune mediators, then signature of inflammation and various inflammatory mediators and other effector immune responses, application of cytokines in therapy and research. Then week 5, there is introduction to innate immunity, how innate immunity influences the whole immunity. Then innate immunity, physical, biochemical, microbial barriers, and pattern recognition receptors and toll-like receptors, that is TLRs. Then coming to week 6 of the innate immunity, we have RLRs and then NLRs and DNA sensors. Week 8 is antigen, a brief introduction, B cells and antibodies, a brief introduction, and then T cells and T cell response, brief introduction. Week 9, will be an introduction on innate immune responses during virus infection and how virus invade innate immunity to establish infection. So this is in two parts, part 1 and part 2. So you will all again have a part 2 of innate immune responses during virus infection and how virus invade innate immunity to establish infection. After that, a brief introduction on B and T cell mediated immune responses during virus infection and how invade adaptive immunity to establish infection. And then coming to week 10, immunological responses during influenza virus infection, part 1, and then immunological responses during influenza virus infection, part 2. Also, a brief introduction of arboviral infection. Week 11 will cover a discussion on immunology of bacterial infections, including immune response to extracellular and intracellular bacteria and evasion of antagonistic mechanisms devised by bacteria to evade such responses. This will also be covered in two parts, part 1 and part 2. After that, we have immunology of tuberculosis, particularly, and this will also be covered in two parts. So, immunology of tuberculosis, part 1 and part 2. Then, in the last and final week, that is week 12, immune responses to parasites and fungal infection, and then a discussion about malaria and African sleeping sickness. So, this is the outline of the course and the syllabus basically of each week and all the topics to be covered during this course. Further information, if anything required, is available on this link, web link. So you can check it out for more information regarding the course. So books and references for this is Essential Immunology by Peter J. Delves, and then Immunobiology, The Immune System in Health and Disease by Charles Janeway, and Cube Immunology, uh, sixth edition or any other higher edition is also very useful. Then other than this, if you want to know the current scenario in research and applications and development of uh, host pathogen interactions and immune system, you can also refer to various reviews and research articles on the particular topic. So you will get the current research that is going on in this field and area. Then infectious disease, pathogenesis, prevention and case studies by Nandini Shetty can also be referred for particular pathogenesis mechanisms and dealing with patient case studies, etc. So all these books and references can be referred for any further details or topics that you might want to delve further into. So this is week 1 session. So here we have history of immunology 1, 2, 3 and 4, which also covers the Nobel Prizes in immunology, the basic introductions and history. And then we have branches of immunology and an introduction of immune system, immunity properties, and an introduction to immune system, link between innate and adaptive immunity, and introduction to immune organs. So these were the topics that were covered in week 1. So the relevant videos are available under the week 1 in the course page. So this session is to clear or to basically understand the assignment that was given after week 1. So we will go into specific topics which will cover each question, and will help you to answer the questions easily and any doubts regarding particular questions. So before going into that, we have to know certain terminologies that are required to easily go through the sessions and the course. So these are the basic terms which are uh, very important to understand every topic, particularly because every time the entire uh, explanation for particular things will not be given. Instead, we'll be using many immunology terms like this. So you have to know what each term specifically means. Only then you can completely understand the sessions. So to delve into this quickly, so first is immunology, of course. It is the study of body's defense against infection. Next is immunity. Immunity is the resistance to any disease. Then immune system. Immune system is the collection of cells, tissues, and molecules that mediate the resistance. So, a collection of the cells, tissues, molecules that mediate this resistance, which is to or against any disease, is by your immune system, and the resistance that it gives is called as immunity. Then immune response. So immunity and immune response, you should understand the difference. The immune response is the coordinated work of the immune system to defend the host against the foreign substances. So response is how it works, how this connection or how this resistance is given, how in coordination many systems and many cells together stimulate a response is your immune response. Then under immunity, again we have types. So the main types are innate immunity and adaptive immunity. So innate immunity is native or it is natural immunity which you already possess, which the host already possesses. Adaptive immunity, on the other hand, is something that you acquire later and develop during a course of infection or during entry of any pathogen. So, adaptive immunity is acquired as an adaptation to infection. That is the difference between innate and adaptive. Innate is something which is already present naturally in the system. Adaptive is something that you acquire to adapt to an infection. Next is antigen. Antigen is a molecule that can bind to an antibody. That is the basic definition, that an antigen is a molecule which binds to an antibody. Then it can be anything, it can be any foreign uh, particle that can bind to an antibody in the host. It can be microbes, it can be drugs, it can be pollen, it can be any kind of allergen, etc. So, or any molecule that can produce an adaptive immune response. So, once your antibody is generated and your antigen and antibody binds, then it will help in acquiring adaptive immunity against that particular antigen. So, an antigen is any molecule that can bind to an antibody or any molecule that can produce adaptive immune response or adaptive immunity. Then antigen determinant. So, antigen will possess an antigen determinant or also called as an epitope. Epitope is a portion of the antigen that is bound to a given antibody or antigen receptor. So, your antigen is a molecule that binds to antibody, but the entire molecule does not bind. A portion of that antigen will be able to bind to the antibody. So the entire molecule is called as antigen, and that particular portion which binds to the antibody is called as the epitope of that antigen or the antigen determinant. So this part is required for the antigen to bind to the antibody. Then antigen presentation. So the process of displaying the antigen as a peptide fragment to the surface of the cell. So an antigen is presented to evoke an immune response in many cases, or to produce antibodies or T cell response in particular. So this is done in several ways. It is done by antigen presenting cells. So how this happens is the or antigen molecule is broken down, and the antigenic part or the peptide part fragment is exposed on the surface of the cells, so that your immune cells or your antibodies can recognize it and bind to it. So it helps in making your antigenic part of your antigen, or the antigen determinant of your antigen, exposed to your immune network. So this process is called antigen presentation. Next is adjuvant. Adjuvant is a killed bacteria or bacterial extracts that can be used to assess the non-immunogenic antigen to induce an immune response. So an antigen can induce an immune response or may not induce an immune response depending on how efficient it is. So sometimes if it can only be antigenic, but it is not enough to produce an immune response. So an adjuvant is such killed bacteria or some extracts which can be which is antigenic in nature but not immunogenic. So this can be used to assess the non-immunogenic antigen to induce an immune response. That is the adjuvant. So these were the basic terminologies required. First is immunology, the study of the body's defense against infection. Second is immunity, that is resistance to the disease. Third is immune system, the collection of cells, tissues, and molecules that mediate the resistance. And fourth is the coordinated work of an immune system to defend the host against foreign substances. Then next is innate immunity, that is the native or natural immunity. Then adaptive immunity, which is an acquired immunity as an adaptation to infection. Next is antigen. It is a molecule that can bind to an antibody, example microbes, pollen, drugs, etc., or any molecule that can produce adaptive immunity. Antigen determinant is an epitope, a portion of an antigen that is bound to a given antibody or an antigen receptor. Next is antigen presentation, process of displaying the antigen as a peptide fragment on the surface of the cell. Adjuvant is a killed bacteria or bacterial extracts used to assess the non-immunogenic antigen to induce an immune response. So these are the terminologies. Yeah. So coming to the topics specific to the assignment questions. First, we will talk about Eli Mechnikov. So, Eli Mechnikov studied phagocytosis, a process by which the cells engulf particles. Okay, this was done in the 1880s, and his work was established, and it was to, elucidate the role of phagocytosis in immunity and inflammation and other tissue homeostasis. So, phagocytosis plays a role not only in immunity but also in tissue homeostasis. What were the observations of Mechnikov? So, Mechnikov observed special cells. So, these were particular type of cells, not all cells do this. So, special cells attacking thorns in starfish larvae. So this work was done in starfish larvae, and he saw that these larvae have specialized cells which were attacking the thorns. He recognized that phagocytosis was a fundamental process that preserved the integrity of the organism. And he proposed that the phagocytosis was a defense mechanism against noxious stimuli. So, any kind of harmful or obnoxious stimuli was fought against by using phagocytosis mechanism. So, this was a part of the defense system. So, you can see here how this phagocytosis is used by the cell. So you can see these are the outline of the cell, that is the membrane, and this is the cytoplasm, this is the nucleus. So you can see how at the periphery of the cell is engulfing the food particle for here. So as we said, it is not only a part of immunity, but it is a part of regular tissue homeostasis. So few prokaryotes, eukaryotes, they have this property to, entrap the food to for a source of energy, and this method they use to entrap this was phagocytosis. So you can see uh, a vacuole kind of structure is formed around the food material, and then it is slowly engulfed within the cell, and then formation of a food vacuole happens. Then the fusion of lysosomes with the food vacuole occurs, because now whatever food has been engulfed has to be broken down and digested. So this lysosomes have several enzymes, and the particular pH helps in the digestion of the food particles. So this is phagocytosis for engulfment of food. Similarly, there is also penocytosis, that is like drinking of the required material. Coming to immunity, how this phagocytosis helps is it helps in engulfing pathogens or antigens. So here you can see an immune cell which is phagocytic in nature, so it's also called a phagocyte. So it has specific receptors, and these receptors help in binding onto the antigen of the pathogen. So once they are bound, the pathogen interacts with the phagocytic receptors, and then the phagocyte envelops the pathogen. Similarly, how the food particles were enveloped here, the pathogen is also enveloped by the phagocytes. Then the cell has also again several enzymes which helps in digestion of this pathogen. One such enzyme is the lysozyme, and then the pathogen breaks down into proteins and other molecules, and then these will now be presented on the surface of the cell. We spoke about the antigen presentation, so that happens here after phagocytosis. Now this cell will present the portion or the peptide of the pathogen which will help in invoking an immune response. So antigen presenting cells. So now an adaptive immunity will be acquired. So this is the phagocytosis. Next is about Emil von Behring serum therapy experiment. So Emil von Behring serum therapy experiment involved injecting animals with a small dose of a small dose of a toxin like diphtheria toxin to induce a immune response or immunity. Then collecting their blood serum, which contained antibodies against that toxin that was injected, and then subsequently using that serum to successfully treat other animals which were infected with the same toxin. So demonstrating the principle of passive immunity transfer through blood serum, essentially laying the foundation for modern serum therapies. So first what he did was he injected an animal with a small dose of the toxin which was enough to uh, invoke a immune response or any sort of immune production, antibody production, etc. Then he collected the serum from that animal and then injected that into a another animal which was also infected with the same toxin, but this toxin would be lethal. So this could uh, cause a disease and eventually kill the animal, but then because of the serum that was transferred into this new animal, the immunity was passive immunity was transferred. So this is foundation to the serum therapy treatments. So therapeutic applications, he then injected the collected serum into other untreated guinea pigs infected with the lethal dose of the diphtheria toxin, observing that the treated animals survived due to the transferred antibodies. Then foundation of the serum therapy, Bering's work established the concept of serum therapy, which uses antibodies from an immune individual to provide passive immunity to another person. So you can see the illustration of the experiment here. Serum from guinea pigs injected with a sublethal dose of diphtheria toxin protects other guinea pigs that are exposed to a lethal dose of the same toxin. So here we see experiment A, B and C. So in A, there is guinea pig number 1 which was exposed to serum from the unexposed animal. So this is your control, and then this uh, serum was given to the guinea pig which was now treated with diphtheria toxin. So this control animal could not develop any immunity because the serum from which it received the animal from which it received the serum was not exposed to the sublethal dose of diphtheria toxin. It was a normal untreated animal. So there is no immunity in this animal which was transferred. So the animal died. But in the next, he first injected the animal 1 with a small dose of the toxin, that is sublethal levels of the toxin, which then invoked immunity in this animal. Then the serum of this animal was transferred to the second animal which was treated with now a lethal dose of the toxin. But this animal survived and there was no disease, there was no diphtheria in this because the the antibodies from the animal 1 was transferred to the second animal, helping the animal to quickly respond to the toxin and eliminate it without establishment of the disease. In experiment 3, he injected a sublethal dose of the toxin from the same bacteria but of a different strain. So, example, first one was strain A, second one was strain B. So, now strain B was given to this, and then the serum from animal exposed to strain B was given to the second animal, and however, the second animal was not exposed to strain B. Was the second animal lethal toxin was strain A. But so this animal also developed diphtheria from strain A and it died. So what this also shows is this is very specific immunity to that particular strain of bacteria. So only if the serum from an animal exposed to strain A is given to another animal which is now infected with strain A toxin, it will help in the passive immune response. However, if a serum from an animal exposed to strain B toxin is given to another animal which is infected with a strain A in a lethal dose, it will not help create an immune response because these antibodies are specific to strain B but not strain A. So the strain A will now establish the infection and the disease will be developed. So the immunity is very specific because the antibodies that were generated were specific to strain A. So it will only further help in preventing the disease of strain A infection. When it is given to treat infections of a different strain of bacteria, then it will not help in protecting the animal. So, it is a specific antibody serum therapy. Next is toxin neutralization by Ehrlich. So, this refers to the concept or developed by Paul Ehrlich that the body produces specific antibodies called antitoxins. It was then called antitoxins, which can bind to and neutralize harmful toxins produced by bacteria, effectively counteracting their poisonous effects. This concept laid the foundation for the understanding of the immune system's response to toxins and paved way for antitoxin therapies like diphtheria antiserum. So this is also a similar experiment. And then one of the most significant applications of Ehrlich's toxin neutralization theory was development of diphtheria antitoxin, where serum from horses immunized with diphtheria toxin was used to treat individuals infected with diphtheria bacteria. So here you have the uh, B cells which will produce antibodies against the exposed toxin or microbes. Now this antibodies can be purified and given to others as an antitoxin to fight against the future infection from the same toxin. So this is antigen antibody neutralization concept. Next is in 1896, bacteriologists Herbert Edward Durham and Max von Gruber discovered agglutination, a reaction that causes the bacteria to clump together. So this discovery is known as Gruber-Durham reaction. So this is the basis of many of the diagnostic tests which involve antigen antibody complex formation. So example, the Vidal test. So the French physician Fernand Vidal used the Gruber-Durham reaction to develop the Vidal test, the first serum test to diagnose an infectious disease. Then later, blood typing is also using the same phenomena. Austrian physician Karl Landsteiner used the agglutination reaction to discover or distinguish the ABO blood groups, which led to the development of blood transfusion. So you can see what is agglutination. The antigen and antibody bind together, form a complex, and then they start agglutinating together in this form of pattern, which helps them to clump together. So this is again a reaction which is concentration specific. So there is a zone of antibody excess, and then there is a zone of equivalence, and then there is a zone of antigen excess. So when there is zone of antibody excess, you do not see the agglutination because the antibody is more than the antigen, so the effectiveness of clumping is not there. And in the zone of equivalence, when they are almost equivalent to each other, you can see that easily they clump together and form this sort of pattern. And when there is more antigen also, it will not help in uh, enough clumping, because excess antigen will also prevent this sort of pattern formation. Then there is also direct agglutination reaction and also indirect agglutination reaction. So in the direct agglutination reaction, a particulate antigen corresponding with its antibody will form a visible agglomerate. So this type of clump together is called as a agglomerate. And how this is a direct agglutination. However, soluble antigen. So when the antigen is soluble, it is sensitized to a particulate, and then it binds to its corresponding antibody to form a visible agglomerate. Because this is particulate, it can easily form the agglomerate by binding to the antibody. If it is soluble antigen, it needs a another particulate to be able to attach to an antibody to form the agglutinations. This is indirect agglutination reaction. A Gruber-Durham-Vidal test refers to a blood test used to diagnose typhoid fever, which is based on the principle of agglutination, where the antibodies in the patient's blood clump together when mixed with specific bacterial antigens of Salmonella typhi, essentially detecting the presence of antibodies against typhoid bacteria, originally described by researchers Gruber and Durham, and later applied to clinical diagnosis by Vidal, and hence a combined term Gruber-Durham-Vidal test. Then mechanism is it detects the antibodies in the patient blood sample that react with the specific antigens present on the Salmonella typhi bacteria, causing the bacteria to clump together. That is formation of the agglomerates. So agglutination is seen when mixed in the laboratory settings. So this is done by serial dilution of the serum, and it is added onto a plate like this, where you add different strains of the Salmonella antigens. You have the typhi, you have paratyphi, ABC, OH, etc. So in each well, the sample will be added, serum, and then the antigen will be added. And based on the dilution, you can also see how much the infection is, how far it is, and the particular strain type will be then identified and diagnosed. Then next is Hans Ernst August Buchner discovered alexin, a substance in the blood serum that kills bacteria. So Buchner named the substance alexin, which comes from the Greek word to ward off. So Buchner was a pioneer immunologist, and he was one of the first to discover a substance in the blood serum that could destroy a bacteria. So Buckner's discovery helped develop the humoral theory, which states that the immunity is conferred by bactericidal and antitoxic substances in the body fluids. So basically, he said that there are substances that are produced in the body and secreted in the fluids, which are bactericidal or toxic to the bacteria, which helps kill the bacteria. So this developed the humoral immunity theory. Then Paul Ehrlich later renamed alexin as the complement. So now we study this alexin as the complement of the complement system, and now we have several components in the complement system, more than 20, which work as a network together to provoke the immune response to kill the bacteria. So complement is now known to be a part of the complement system, which is made up of 20 proteins that work together to eliminate the infectious organism from the body. So we have different pathways again in the complement system. We have classical pathway, we have the alternative pathway, we have the lectin pathway. So the classical pathway, I'll just basic intro I will give. So this will be later explained in the coming up sessions. So the classical pathway, we have the components C1, C4, C2, C3, C5, and then the membrane attack complex, which is made up of C5, 6, 7, 8, and 9. So basically, the C1q binds to the antigen over here, and then you have the C4 and C2, which splits into C2b, C4b, C2a. So the C4b and C2a then form a complex, forming the C3 convertase. So then it hydrolyzes the C3, and it splits into C3a and C3b. C3b helps in phagocytosis, opsonization. C3a is chemoattractant, and it is involved in inflammation. Then C3b then further binds to the C4A, C2A complex, forming now a C5 convertase complex. So now the C5 convertase will hydrolyze the C5 component, which will again split into 5A, which is again uh, in inflammation, and C5b will now bind to C6, C7, C8, and a multimer of C9, which will help in pore formation on the target cell and leading to osmotic imbalance and finally cell lysis. So this complex is called as membrane attack complex. So this is the classical pathway. Other than this, we also have alternative pathway where it directly starts with C3 with spontaneous hydrolysis of C3, and then C3 then finds the binds to factor B, C3b, and factor BB will now become the C3 convertase here and again hydrolyze C3 into 3A and 3B. Then again same C3b will form a C5 convertase with C3BB and C3B factor BB. And now this C5 convertase will again hydrolyze C5, and C5b will form complex with 6, 7, 8 and 9 and form a membrane attack complex. This is the alternative pathway. Other than this, we also have the lectin pathway, which is a mannose binding lectin, which binds to the carbohydrate groups of the bacteria or the pathogen. And then directly we again have C4, C4A, C4B, and in a similar way we again have C3, C5 and a membrane attack complex. So the trigger point is varying in all, but the end is the membrane attack complex formation, which helps in uh, clearance of the pathogen or the infected cells. Next is the key observation in Pfeiffer phenomena. So in Pfeiffer phenomena, when the bacteria are injected into an animal previously immunized against those bacteria, the bacteria rapidly disappears from the body due to destruction by the immune system. Even though no visible lysis or cell breakdown is observed in the test tube while mixing the bacteria with the immune serum, this indicates that the release of toxins from the bacteria upon interactions with the antibodies, which was groundbreaking discovery in understanding bacterial pathogenesis. So in vivo observation was the crucial observation was made in living animals where the bacteria were rapidly destroyed within the cell within the body after injection, not just in the test tube. Then bacteriolysis without visible cell lysis. So although bacteria appeared to dissolve in the body, direct observation in the test tube did not show obvious cell lysis, leading to the concept of endotoxins that were released from the bacteria. So this phenomena then helped establish the importance of immune system in combating bacterial infections and providing insights into the mechanism of antibody mediated bacterial killing. So this is basically antibody mediated bacterial killing by Pfeiffer. Next is Edward Jenner. So Edward Jenner is for the vaccines. So the famous cowpox that was used to develop a vaccine uh, in an 8-year-old boy. So Edward Jenner used cowpox to develop the first successful vaccine. He inoculated an 8-year-old boy with the matter from a cowpox sore or a cowpox pustule on the milkmaid's hand in 1796, and this method is called arm to arm inoculation. Jenner was an English physician who noticed that people with cowpox were immune to smallpox. Not only that, he also saw that people who are immune, who had this cowpox pustules but did not develop any disease, they also were immune to the smallpox, which was actually affecting the humans and was a epidemic or a pandemic that was killing a lot of people at that time. So he used the cowpox instead of the smallpox because it was less dangerous and Jenner's discovery led to the era of preventative measures for contagious diseases. So Jenner is often called as a father of vaccines because he used a scientific approach to prove the method worked. And the boy Jenner inoculated was James Phipps. He felt unwell for a few days after inoculation but then made a complete recovery. So, 2 months later, Jenner inoculated Phipps with the matter from smallpox sore to test his resistance. And Phipps remained healthy and became the first person to be vaccinated against the smallpox. Then, now we have theories of immunity. We have cell mediated immunity and humoral theory of immunity. So, the cellular theory of immunity focuses on the direct attack of the infected cells by immune cells like T lymphocytes. However, the other school of thought was the humoral theory of immunity, because we saw that how no visible cell lysis was seen in case of Pfeiffer uh, here. So even though there was destruction of the bacteria, the cell lysis was not seen. So then humoral theory came up saying that it involves production of antibodies by B cells which circulate in the bodily fluids to neutralize pathogens outside of the cells. Essentially, both of them occur in the body. So cellular immunity targets intracellular pathogens and humoral immunity targets the extracellular pathogens. So the major uh, points or the major characteristics of cell mediated and humoral immunity are: cell mediated is mediated by T cells and humoral is by the B cells. In cell mediated, we can see no formation of antibodies. Humoral is mediated by formation of antibodies. Cell mediated secretes lymphokines, and here we do not see any lymphokines. Cell mediated receptors are used to detect the antigens. Here antibodies are directly detecting the antigens. In cell mediated, T cell receptors bind to the T cells and the T cell receptors bind to the T cell and then they bind to the antigens. Uh, T cell receptors present on the T cell bind to the antigens, and here the antibodies produced by the B cells bind to the antigen. Then T cells, that is cell mediated, it protects against virus, fungi, and other intracellular bacterial pathogens. Humoral, however, protects against extracellular virus and bacteria. Cell mediated immunity, it eliminates the tumor cells and also protects against cancers. And humoral does not really play a role in tumor cell elimination. In T cell response, both CD4 plus and CD8 plus cells are involved in the cell mediated immunity, and in humoral immunity, only the Th cells, that is the T helper cells, are involved in the humoral immunity. Cell mediated, it mediates hypersensitivity type 4, and humoral can mediate hypersensitivity type 1, 2, and 3. Cell mediated immunity, it shows delayed response. However, the humoral immunity is a quick response. Let me just connect the charger. Yeah. So cell mediated is a delayed response and humoral is a quick response. So these are the basic characteristics of cell mediated and humoral immunity. Yeah. So the next is innate immunity. It is the body's first line of defense against harmful substances. It is present at birth, it is native to the host, and it lasts the person's entire life. Innate immunity includes physical barriers, immune cells, and proteins. Physical barriers include skin, the first line of defense against infectious agents, mucous membrane found in the gastrointestinal tract, sinuses, and lungs, tears in the flush, they flush the eyes to prevent the infection, And stomach acid, it helps keep the harmful substances out. Then the immune cells, neutrophils, the most common white blood cell in the bloodstream. And monocytes, they develop into macrophages upon activation. Then eosinophils target the foreign cells that are too large to inject parasites. And natural killer cells, part of the innate immunity, and mast cells are also part of the innate immunity. So here we can see the physical barriers, skin, the gut, and the lungs. And then the chemical barriers, the stomach acid, lysozymes in the eyes. Here we have galt and malt. And in the innate immune response, we have inflammatory cells, mast cells, neutrophils, macrophages, natural killer cells. Then we have adaptive immune system. In adaptive, we have cell-mediated response and humoral response. Cell-mediated and humoral we just now discussed, and then we have T lymphocytes, and here we have B lymphocytes, T lymphocytes CD4, CD8 plus, and here we have production of antibodies. Then we also have dendritic cells, which are a connection between both innate and adaptive immunity. They can give, they are involved in both types of immunity. Then the regulation of immune responses involves a complex system of checks and balances, primarily controlled by specialized immune cells like T cells and regulatory molecules, which ensure the body mounts an appropriate immune response against the pathogens while preventing excessive inflammation or attacks on its own tissues. With factors like nature of the antigen, its dose, and route of administration playing a key role in determining the response level. So, innate immunity recognition, the initial detection of the pathogens by immune cells, which triggers the first line of defense through the mechanism like phagocytosis. Then we have adaptive immune response, a more specific response involving B cells and T cells, which produce then antibodies, and T cells directly attack the infected cells, activating the specific antigen that is encountered, the specific response is activated. Then we also have Tregs, which are the T regulatory cells, a type of T cell that actively suppresses excessive immune response, preventing autoimmune reaction where the body attacks its own tissue. Once the immune response is triggered, there should always be a control, there should always be a uh, check to the response. You cannot leave it unsupervised or unchecked if it is, going past a threshold. So when that happens, it will start attacking your own cells and cause self harm, and it will cause damage to the healthy cells and tissues. So we have Tregs for this purpose, which helps regulate the response, and it will suppress the immune response once the pathogen clearance is done, or once the required amount of immunity has been generated. Then we have cytokines, signaling molecules produced by immune cells that can either promote or dampen the immune response depending on the type. So these are involved in both pro and anti-inflammatory. So depending on the type of cytokines that is produced, they can either be pro-inflammatory, that it can promote the immune response, or it can be anti-inflammatory, it can suppress the immune response. Then we have immunological tolerance, the ability of immune system to recognize and not attack self antigen, preventing autoimmune disease. So the immune cells are always selected on the basis that they can distinguish between self and non-self. If they are unable to make this kind of distinguishing, if they are unable to distinguish such differences, then the self damage can occur. It will create autoimmune diseases. So the immune tolerance is very crucial. Then immune checkpoints, molecules on the immune cells that acts as brakes on the immune response, which can be targeted in cancer immunotherapy to enhance anti-tumor activity. So the immune checkpoints hold the immune response or stop the immune response at particular times. But this can be targeted in cancer therapy to enhance the immune response or amplify the immune response in anti-tumor immune activity. Then factors influencing immune response regulation. Antigen type and dose influences the characteristics of the invading pathogen, such as its protein structure, concentration, determine the strength of immune response that is generated and the type of immune response that is generated. Then route of antigen administration or route of antigen exposure also regulates the immune response, and how the body encounters an antigen, whether it is through the skin, through the mucosal surface, through an open wound, or the inhalation or anything, uh, that particular type of immune response will be given to it, and it can influence the way your body will now deal with the antigen. Then tissue microenvironment. The local conditions of the tissue can impact the immune cell behavior and the function. Then age. Obviously, with age, the immunity will vary and it will get weaken. So immune function can decline with age, leading to a weaker response to infections. Then stress and hormones. Chronic stress suppresses immune function, while hormones can modulate immune responses. So that is it for the session 1, all the topics that were covered based on the questions. So if there are any other doubts to be cleared regarding this session or any other session, you can mail me on this mail ID, or you can post it on the NPTEL page or enquiry page, which will be discussed in the further sessions. Thank you. We can end the session here.